Hello, let's move on to the next part of the chapter. We'll be learning about the other cell organelles. Now that we have learned about cytoplasm, nucleus and a few other cell organelles, let's dive on into other cell organelles such as the mitochondria, plastids, vacuoles. I know there are a lot of cell components, but slowly, and like I have been telling you, if you visualize all these structures, if you visualize the location of all these structures, you will be easily be able to remember the functions. Let's get started on with this part. First of all, we are having here the mitochondria. Okay, mitochondria is very famously called the powerhouse of the cell because there is some important activity that is going on over here. What is that activity? That activity is that all the chemical initiation that takes place in a cell, the mitochondria is that site where that chemical initiation is taking place. Okay, what I mean here is that it is responsible for creating the chemical energy of power to power the biochemical reaction in the cells of most eukaryotic organisms, since they are the site of cellular respiration and storage centers of ATP, the energy currency of the cell. They are known as the powerhouse of the cell. What we mean here by this is that there is some chemical reaction that goes on inside our bodies, right? These are known as the biochemical reactions. Now, when I say that mitochondria is the site where these chemical reactions they are initiating, okay, means some energy, some chemical energy, it has to be produced at first somewhere so that the other processes they may be carried out smoothly. All right, so mitochondria is that site where chemical energy is produced, is created to power the biochemical reactions in the cells of most eukaryotic organisms. So, mostly mitochondria is that site where this initiation happens. Initiation of what? Initiation of chemical energy. Why? So that biochemical reactions may occur inside our cell. Biochemical reaction which is important for our survival, for our basic metabolic functions. And since they are the site of cellular respiration, mitochondria, they are the site of cellular respiration. Now, we humans, for example, we breathe in through our nose and we breathe out also through our nose. But the respiration, this is just the breathing process. The actual respiration is taking place inside the cell. The actual respiration means the actual, inhalation, the absorption of oxygen and the giving out of carbon dioxide, that process of respiration that takes place at a cellular level, and inside the cell mitochondria is that organelle, that cellular organelle where this process takes place. And also this chemical energy that is being produced inside the mitochondria, it is stored in the form of ATP. ATP, you may call it as the energy currency. Okay, so here if you may see in this diagram, ATP synthase, this is the site where this ATP, this energy currency is being created so that it may fuel, it may be able to engender, cause all the other biochemical reactions that are occurring inside the cell. This is the mitochondrial DNA. Interestingly, mitochondria has its own DNA. All right, DNA, like I had told you, the hereditary, all those components that have this hereditary code embedded in them. Okay, so interestingly, mitochondria has its own DNA, has its own hereditary code. This is the this is the matrix granule. There are also some ribosomes that are found inside mitochondria, and observe this structure. This is a very curvaceous finger-like structure, right? This structure is known as cristae. Okay, this cristae, and this cristae has a membrane of its own, which is known as the crystal membrane. Okay, this cristae has a membrane of its own, which is known as the crystal membrane. And then a mitochondria is double layered structure. It has an inner membrane, inner boundary membrane, and it has an outer boundary membrane. Okay, and between the two folds of the cristae, there is some intra crystal space, there is some peripheral space, and there is some other tubular rounder structures, round structures, not tubular, I'm sorry, which are known as parents. Okay, so all in all, you have to remember that mitochondria is the powerhouse of the cell. That is important because the energy, chemical energy is initiated here, which powers, which fuels, which engenders all the other biochemical reactions that are happening inside the cell. It is a double membrane bound organelle found in most eukaryotic organisms. Also, it is rod shaped and membrane bound, that that cristae that is curvaceous, that is rod shaped, or you can also say that finger like shape, okay, with an intermembrane space of its own in between this intercrystal space. Let's move on to the plastids. Plastids are those cell organelles. They are found in plants. Okay, they are responsible for the colors which you observe in plants. The green color, or the fruits color. That some may be red, some may be yellow, some may be orange. So that color is owing to these cell organelles, these plastids. Okay. The basic structure of plastids over here, you can see there is also, these are also double membrane structure. There is the outer membrane, and then you have over here the inner membrane. There is the intermembrane space. The liquid inside, or you may say the jelly-like matrix inside the plastids, is known as stroma. It is an aqueous fluid. Okay, so it does not have much of a solid-like structure. It is more of a liquid-like structure. So it is an aqueous fluid, and inside this plastids you have another disc-like structures, circular solid disc-like structures. What are these? These are your granum. Okay, and granum, these are stacks of your thylakoids. So this individual disc-like structure over here, this individual disc-like structure is called your thylakoid. Okay, and when say 2-3 pairs of thylakoids, they are stacked together, one granum is formed. All right, and as you can see here in the diagram, there are many granums, right? So plural of granum is grana. Okay, plural of granum is grana, and all these individual stacks of thylakoids, all these individual granums or grana, they are interconnected through each other by this fibrous sort of material, which is the lamella. Okay, and you have a pigment inside of the thylakoid that we called as the lumen. Okay, so this is your structure of plastids. Plastids is a double membrane bound organelles that are mostly found in plants and some algae, since algae also have distinct color. All right, they may be spherical, ovoid, or discoid. That may be a spherical or ovoid or discoid in shape in higher plants, and they may be stellate, cup shaped, or even spiral in some algae. So it depends on which organism you're talking about, and that shape may vary to spherical, ovoid, discoid to stellate, cup shaped, or even spiral. And inner membrane encloses matrix called stroma. Okay, so you can visualize stroma to be the own cytoplasm of plastid. Like nucleus had that nucleoplasm, so plastids will have the stroma, which contain grana. Function, they are responsible for the plant's metabolic activities, and they are of 3 types: chloroplast, chromoplast, leucoplast. Chromoplasts, if we have to talk about it, that important point that I had told you, that plastids are responsible for giving plants, fruits, flowers their specific colors. So chromoplasts are those plastids which give the fruits and the flowers their colors. So those colors may be anything except for green. Okay, because green in plants, it is the indication that chlorophyll is present. And by the name over here, chloroplast contains a pigment, green pigment, and that green pigment is none other than chlorophyll. Okay, so chromoplast also had pigments. It is also responsible for the pigmentation of flowers and fruits, but those pigments could be any other pigment except for green. If a chromoplast has green pigment, then that chromoplast is called as chloroplast. It contains chlorophyll, and that then naturally takes part in the process of photosynthesis. Whereas leucoplasts, they have no color, and a common function of all the plastids is that they help in the storage of food. All right, so chloroplast also does that, chromoplast also does that, in addition to those colored function. But leucoplast, since it has no color, so it is essentially involved in the storage of food and storage of other components that the cell may require for its metabolic survival. Now let's learn about vacuoles. Vacuoles are found in both plants and animals, but usually vacuoles are found more in plants. Alright, there is cell wall, there is the cell membrane, nucleus, ribosomes, mitochondria, and endoplasmic reticulum. This is the plant cell, since we have cell wall over here, and you see there is such a big vacuole. So because plants, the most important function of plants is that they store food. All right, so they need lots of spaces for the storage of this food, and as you can see, vacuole is taking up a lot of the space of the plant. So the essential function of vacuole is the storage of food. Okay, vacuole is a membrane bound organelle containing liquids or solids, because food may be stored in any other form. Property is that these are permanent and large in plant cells. Plant cells have to have vacuoles inside them. They cover almost 90% of the cell space in mature plants. As you can see over here, this vacuole, it's covering so much. Even the nucleus is not so big as is the vacuole. All right, so vacuoles take up space as much as 90%, and their main function is to ensure the rigidity of the cell and maintenance of its osmotic pressure, but it is also responsible for the storage of food. All right, because of its large size, it ensures that the cell is rigid, and the osmotic pressure, that means that pressure that the cell exerts towards its cell wall. All right, in case of being put into a hypotonic solution. Okay, so in that case also, vacuoles, they perform the function of maintaining its osmotic pressure. Can you name two organelles we have studied that contain their own genetic material? If you may remember, mitochondria, there was that mitochondrial DNA also in that diagram. Mitochondria is one cell organelle that has its own genetic material. Besides mitochondria, what is the other organelle? The other organelle is the plastids. Plastids also have genetic material of their own. If organization of cell is destroyed due to some physical or chemical influence, what will happen? Now, if any physical injury were to happen, or if any chemical influence were to happen, and if an organization of a cell is destroyed, then basically there will be no metabolic activity, right? There will be no respiration, there will be no, say, production of food or any growth. Then how will that cell be able to function, right? Naturally, that cell is gonna die, right? That cell is gonna die, and no living function is going to take place due to that cell. So when an organization of a cell is destroyed due to physical or chemical influence, that cell dies because there will be no metabolic activities, because there will be no living function happening inside the cell. Why are lysosomes known as suicide bags? So if you may remember from one of the previous slides, that lysosomes, they have the special ability of destroying any foreign particles such as bacteria, virus. So if bacteria or virus were to intrude the cell membrane and come inside of the cell and harm the cell components, so what lysosomes do? They secrete some secretions that can destroy bacteria and virus, but in the process they sacrifice themselves. So because they are killing the harmful foreign components, but they are also dying in the process, lysosomes are known as suicide bags. Let's learn about ribosomes now. Description is that these are small particles that contain chemical messengers called RNA. RNA is a ribonucleic acid. Your DNA, those hereditary compounds, they are basically made up of RNA. And ribosomes are those organelles where protein synthesis happens. This is a very important function. So ribosomes are small particles that contain chemical messengers called RNA, and ribosomes are flattened, and mostly they are spherical in shape and have two major subunits. Function is that they play a major role in protein synthesis. You have to remember this. This is the site of protein synthesis. And their presence in ER gives rise to rough endoplasmic reticulum. This I told you while I was explaining you endoplasmic reticulum. That endoplasmic reticulum, when they have ribosomes attached to it, they are known as rough endoplasmic reticulum. The structure of a ribosome is somewhat like this. There is two units, right? So there is one larger subunit and there is one smaller subunit, and there is a central protuberance, a central, rising part. All right. And this is your structure of ribosomes, where are proteins synthesized inside the cell. Proteins are synthesized inside the cell of ribosomes. All right. Ribosomes are the site of protein synthesis in cell. Let's learn about centrosomes. Centrosomes are unbounded organelles, and they are only found in eukaryotic cells of animals, and they contain centrioles. They are only found in eukaryotic cells of animals because they essentially are involved in the growth. They are essentially involved in the cell division process. So, since cell division process is different in different organisms, so therefore centrosomes, they are mostly and only found in eukaryotic cells of animals. Property is that centrioles, they form two barrel-shaped clusters. As you can see here in this diagram, this barrel-shaped structure, they have another, inside this structure they have many small tubules, many small interconnecting fibers of microtubules. Centrioles form two barrel-shaped clusters of microtubules, and they're linked together by another small interconnecting fibers. And these are the cylindrical microtubules that help in the process of cell division. Now that we have talked about the components of the cell, let us learn about the cell division, where we'll be learning about two important processes. One is mitosis and the other is meiosis. Let's have a look at the process of cell division. New cells are formed in organisms in order to grow, replace old, dead and injured cells, and to form gametes required for reproduction. The process by which new cells are made is called cell division. And the cell division is mainly of two types. We have mitosis and we have meiosis. Mitosis is the process of cell division by which most of the plants divide a cell for the growth. Okay, so the plant cells or the animal cells, they may die of injury, or they may die of some other, abrasion. So when that happens, the cells need to multiply. Cell needs to divide and grow again, so that the old or the dead or even the injured cells, they are replaced. This process of just replacing older cells by newer cells is known as mitosis, and in the case of mitosis, each cell is a mother cell. By this we mean that there is supposed to be one cell, alright, in this you will see in meiosis, there's supposed to be some specific mother cells, and from those specific mother cells, new cells would be formed. But since over here the motive is just to replace the old or the dead or the injured cells, so each cell acts as a mother cell, and from those cells we get two identical daughter cells. Alright, so one cell leads to the growth or leads to the production of two cells. The daughter cells have the same number of chromosomes as mother cell. It helps in growth and repair of tissues in organisms. Number of chromosomes in mitosis remains the same. This is very important to remember. This is the pictorial representation of how a mitosis takes place. This is a mother cell. This is, let's just say, is a normal cell. Now it has, let's say, this chromosome, okay, red chromosome, and this has this white chromosome. Okay, so DNA replication is happening. That is why in order chromosomes to get divided, these will be replicated. The DNA, that component, hereditary code that are present inside the chromosomes, they are replicated, and then, as you can see in the second picture over here, a scissor sort of, or a sort of X-like figure, or a scissor sort of structure is being formed. When that happens, now another phase occurs in the mitosis process, which is known as the metaphase. In this, what happens? What happens? Centrosome comes in. All right. Centrosome, they comes into the nuclear membrane. During cell division, what happens? The nuclear membrane, it dissolves. Okay, so that this centrosome can come inside and it can facilitate the process of the separating of the chromosomes. So what happens? Centrosome comes in, and owing to their tubular microfiber sort of structure, these structures, spindle shaped structures, they occur. And what happens is that these spindle shaped structures, as you can see that the cell has expanded, the spindle shaped structures, they have been formed, and the two chromosomes, the duplicated chromosomes, they are held, and then during the cell division phase, they are separated from each other. So this replicated scissor-like figure over here, this is all this two parts, this replicated, right? So this is going to be broken up now. This is going to be divided now. Same goes for the other chromosome over here. So both of these chromosomes, the part of these chromosomes, they are breaking up with the help of the micro fibers or microtubules of these centrosome after the metaphase. And what happens is that since they are now broken up, a cell before the division was occurring had one pair of chromosomes. One, you can say, for the sake of simplification over here, one is this red chromosome, one is this white chromosome. So when this breaking up happens, final stage, when we come to this cell breaks into two parts, all right? So each part will have now that red chromosome and that white chromosome. So since this duplicated pair has been broken off, has been snapped, has been snatched away, or has been divided by these spindle shaped structure, so now we can easily say that mitosis has been completed, and the number of chromosomes that we had in the mother cell, we have those chromosomes in the daughter cells as well. Now that we know what mitosis is, let's learn what meiosis is. Specific cells of reproductive organs or tissues in animals and plants, they divide to form gametes, which after fertilization give rise to offspring. So some specific, now mitosis, that was only for the cell division, right? If the cell is, say, injured, or if the cell is old. But if a new organism is to be formed, then that cell division, it occurs by the process of meiosis. So in meiosis, there are some specific cells only, which are the mother cells, and those specific cells are found in the reproductive organs or reproductive tissues, be it in animals or be it in plants. And those reproductive cells, they have this special name of gametes, okay? And gametes of two individuals, they combine together, and that combination process is known as fertilization, and then there is this rise of an offspring. All right, so this meiosis cannot happen in one say organism on its own. Mitosis can happen in one organism on its own, but for a meiosis to take place, one gamete has to come from another organism. So yes, the process may happen in one individual, in like humans, the meiosis process, it does happen, in both male and female, but the fertilization process happens in the female. All right, the process of meiosis, so that the gametes, so that those specific cells, the reproductive cells and reproductive organs, they are produced. Okay, this process is called meiosis, and it involves two consecutive divisions. The difference here is that in meiosis, the number of chromosomes is halved. Okay, so when a cell divides by meiosis, it produced 4 new cells. So one cell produced 4 new cells, and instead of just 2. All right, so mitosis had one cell giving rise to 2, but in meiosis, one cell is giving rise to 4. And can you think as to why the chromosome number is reduced to half in the daughter cells? It is reduced to half because when two gametes, when they can fertilize, then that half chromosome and the other half chromosome from the other gamete, and that gamete, remember, is coming from another organism, then that can give to one chromosome, that can lead to one chromosome. So when you say that that same old hereditary example, that you look like your mother, you look like your father, it's because you have some specific DNA coding that includes both of your mother as well. Similarly, the same thing is happening in plants as well. The genetic coding is halved. All right, one comes from one gamete, say from one parent, say the father, and the other is coming from the other, say the mother. So when these two halves are combining, they are coming together, these cells, when they are fertilizing, then you get one whole chromosome. And the process of meiosis can be depicted here. This is the mother cell. All right, so chromosome is duplicated, the same X-shaped or say the scissor-shaped structure has been formed. All right. Then comes the cell division phase. You have those spindle-shaped, structures again appearing from the poles of the cell. Always remember, they appear from the poles of the cell, from both the ends. All right, they are occurring and they are again causing a snapping or breaking of these chromosomes in the middle. Okay, but in the first phase, they are not divided into two halves. In the first phase, what is happening is, yes, they are dividing. Say, if this is one chromosome, okay, wait. If this is one chromosome, all right, one X-shaped chromosome, and if this the other X-shaped chromosome, and they are bounded by these fiber-like structure from the poles of the cell. All right. So in the first phase of the cell division, which we also call as the the meiosis 1, in this phase, they are not separating into two two other distinct chromosomes that they were in the case of mitosis. But what is happening here? They are separating into two, in the process of this meiosis 1, but one part of this, it is being transferred to this part. All right, and one part of this chromosome, this pair of chromosome, is being transferred to this part. Okay, and then they are again combining together. Together. I'm so sorry for my drawing. It's not, it may be not that meaningful in what pictures I'm drawing, but I hope you're getting the concept, because the understanding of the concept is important. All right, I'll be explaining you here once again. So let's just say here that when this middle shaped structure they appear, okay, so this is that stage. All right, this is the meiosis 1. Okay, in this stage, what is happening is that they are not immediately breaking up into two particles. This X-shaped figure, if you remember mitosis, what happened? One particle used to go here, the other particle used to go here, and two daughter cells are formed. All right, here that is not happening. Here what is happening is that this one part of this chromosome, all right, it is being transferred to this part. That is why over here in this picture, see this picture over here and the bottom, the white scissor-like structure, it has one red part, and the red figure-like structure, scissor-like structure, it has one white part. So one exchange of genetic code, exchange of DNA is taking place, and when that happens, you get two cells. Okay, so you get two cells over here, somewhat like the mitosis thing. All right, but this is not where meiosis is gonna end. There is going to be another phase. In another phase, all right, these two cells is going to that process individually, where you have this red shaped part has one white part of the chromosome, and this white shaped chromosome part has one red part. All right, so this is going another phase of separation. So another time these fiber like structure is gonna occur from the poles of the cells, and now they will try to break this connection, this scissor like connection in the middle. Now they will try to break this connection over here, and now there will be one chromosome over here that has that has this white part with the red part. Then there is this another chromosome over here that is just the red part, and for the white one also, here you have just the white part, and you have the white part with the red part. So basically what is happening is that some DNA is getting exchanged. Okay, some processes, they are getting exchanged, and when that happens, a final division occurs, and during that final division, the number of chromosomes, they are halved. So the mother cell had two chromosomes, the four daughter cells will have one chromosome each, and there will be two cells in which the chromosome would be intact, as in there were two chromosomes, there will be only one chromosome, the number of chromosomes will be halved, but the nature of the genetic coding, that will be intact. But in two of the daughter cells, there will be one exchange of some information. Okay, this is the specific way in how gametes are being divided. Okay, how gametes are basically formed, and the whole process of meiosis is gonna come clearer to you when you will learn about fertilization. So now you may be confused why this genetic exchange is happening, but when in the further chapters you'll be learning about fertilization, this process will be very much clear. So this is all that you had to learn here in this chapter, cell, the fundamental unit of life. Now let's come to a quick revision. What all we have learned in this chapter? We have learned that cell is a fundamental unit of life. We have learned that there are different, different types of cell. There is a prokaryotic cell. Then there is a eukaryotic cell. Then there is also unicellular organisms. There are multicellular organisms. There are so many things that we have covered. We have also had a look at the number of cell organelles, the functions, the structure of these individual cell organelles, and we also had a chance to look at the cell division. So let's come to a quick revision. The fundamental organizational unit of life is the cell. Cells are enclosed by a plasma membrane composed of lipids. It is composed mainly of cellulose, and it is located outside the cell membrane, whereas the cell membrane is the active part of the cell. The cell wall is the dead part of the cell, is the non-living part of the cell. And the cell membrane, it regulates the movement of materials between the ordered interior of the cell and the outer environment. That means that the cell membrane is semi-permeable in nature. It only allows some specific material to be taken inside of the cell and to be taken outside of the cell. That is because it protects the cell from its outside environment, whereas the cell wall is freely permeable. The presence of the cell wall enables the cells of plants and fungi and bacteria to exist in hypotonic media without bursting. If you remember, this is because when the water has has reached its to its maximum content inside the cell, then the cell is gonna exert osmotic pressure outside. All right, because that pressure is basically because of the extra solvent molecules that have entered inside the cell. But the cell wall, it's gonna exert an equal amount of pressure inside the cell, and that pressure that this cell wall exerts inside the cell is gonna prevent the cell from bursting. The nucleus in eukaryotes is separated from the cytoplasm by double-layered membrane, and it directs the life processes of the cell, like synthesis of protein, like the cell division, etc. Endoplasmic reticulum ER functions both as a passageway for intracellular transport and as a manufacturing surface. So the endoplasmic reticulum, it functions both as a passageway for intracellular transport as well as a manufacturing surface. Golgi apparatus, which have which are composed of cisternae, and they also have vesicles. All right, those sacs-like structure. They consist of sacs of membrane-bound organelles. Golgi apparatus is a double membrane structure. Vesicles that function in the storage, modification, and packaging of substances manufactured in the cell. Most plant cells have large membranous organelles called plastids, and plastids are mainly of two types: chromoplasts and leucoplasts. So chromoplasts, all the plastids, chromoplasts, leucoplasts, also a special kind of plastids which are known as the chloroplasts. All of these are involved in the storage of food. But chloroplasts have this extra function that they incorporate the colored particles to the to plant components such as the flowers, the flowers, and then there is the fruits. So any other color except for green, because when the green color occurs, it is an indication that that plastid has chlorophyll, and that chlorophyll is responsible for photosynthesis. So chromoplasts that contain chlorophyll are called chloroplasts, and they perform photosynthesis, whereas leucoplasts have no color, they do not embody any other color, and their main function is to just store the food that is being produced by plant. The primary function of leucoplast is storage. Most mature plant cells have a large central vacuole that covers around 90%. of the whole cell that helps to maintain the turgidity of the cell and stores important substances including wastes. Prokaryotic cells, they are no membrane bound organelles. They have no distinct nucleus. There is no nucleolus present. Rather than that, they have only nucleic acid. So their chromosomes are composed of only nucleic acid, only nucleoid, and they have only small ribosomes as organelles. No other membrane bound organelles in the prokaryotic cells. Cells and organisms, they divide for growth of body, for replacing dead cells, and for gametes for reproduction. For replacing the dead cells or the injured cells, we have the process of mitosis, and for producing gametes for reproduction, we have the process of meiosis. So we have these two different kinds of cell division. This is all that we had to learn here. We had learned about so many different organelles. We also learned about lysosomes, ribosomes, and different organelles. Remember, it's very difficult to remember all the functions at once, but if you remember to visualize how a certain organelle is shaped, like how a certain organelle is located, where it is located, then you may be able to figure out its important functions. Okay, so this may be a lot to take in, but revise, go through the chapter, go through the recordings, and I hope that I have been of immense help to you in figuring out where these different organelles are located, what are their structure, what are their functions, and I hope that it is clear to you why cell is called the fundamental unit of life, how the different life processes that occur inside our body is being carried out at that microscopic cellular level, and I hope that the differences between some of the plant cells and some of the animal cells is clear to you. So thank you so much for being with me here today. Take care and study well, and stay tuned on Study Room for more chapters, be it in biology, chemistry, physics, and rest of the subjects. So take care and study well.